Okay, welcome to our second lesson of chapter 2. We are going to be looking at our microscope. In our previous lesson, we looked at the different parts of a cell, the different types of cells, and how plant cells and animal cells might be different from each other. In this particular lesson, we're going to learn how we know that stuff. We know that because we can look through a microscope and see all of those different components, or most of them at least. So in this particular lesson, I'm going to ask you to do some work before you come back and listen to the rest of this lesson. Go to page 605 in your textbook. It's provided on the classroom if you need it. And I want you to fill in the parts of the microscope. They're listed exactly as you see here. And I also want you to write down the function of them as well. It might be a good idea to put it into your own words before you write it down. That way, one, you have less information to write it, and two, you actually understand it before you write it down, right? So I'm going to ask you to write that down and then come back to this point of the lesson so that you can listen to the rest of it. This slide is the reason why I wanted to record over and talk through this lesson is because of the magnification. So knowing the parts and the function only takes you so far. If you're using a microscope, you will have to use the different components of it. But I might ask you a question. For example, I might ask you, how zoomed in are you when you're looking at a microscope, right? So the microscope allows us to see things that are very, very small because you're zooming in. But to what degree? We can calculate that based off of the information that we have about the microscope. This is what we call magnification, right? So if you're looking at something that's very, very small, you're zooming in, you're magnifying it. Every part of the microscope has a different level of magnification. So the first thing that I want to talk about is your eyepiece or your ocular lens. So it can go by either name. And the top part of this is where we are going to be looking through. So you look through this particular part of the ocular lens or the eyepiece. It can go by either name. And this section right here has a 10-time magnification. If you're looking at just through your ocular lens, you will see things 10 times bigger. Okay, the second part of the microscope that I want you to, I want to talk about is this section right here. So this is where the rest of our lenses are. And it's held on by this section right here, which is our revolving nose piece. That allows you to turn between the different objective lenses that are there. And in this picture, it's hard for you to tell which is low, which is medium, which is high. But in person, it's actually quite easy because the smallest one is low, the medium one is medium, and the longest one is high. Each of these different lenses has a different level of magnification. So the low one has 4 times, the medium one is 10 times, and the high power one is 40 times. So just for now, I just want you to know the magnification of each of these things individually. If you're looking through the low power objective and nothing else, not even the eyepiece, you will see things 4 times closer. However, these parts are obviously connected in a microscope, right? So how do we figure out the total magnification of what you're looking at through the microscope? We have to use this particular formula right here. Okay, so you're taking the magnification of the ocular lens power, you're multiplying it by the magnification of your objective lens power. Your objective lens can be one of 3 things. It can be low, medium, or high, and each of those has a different magnification, just like we talked about. So if you're finding the total magnification of a microscope and it's on the low setting, you have your 10 times from your ocular power, and you're going to multiply it by your 4 times, which is our objective lens power. This gives you a total magnification of 40 times. Same things for the other 2 objective lenses as well. So if you're on a medium setting, you have 100 times total magnification. If you're on the high setting, you've got 400 times magnification. So that's pretty much what I want you to understand about about the total magnification of a microscope. It's different for each objective lens power that you're at. Okay, and the next thing that I want you to look at is to flip in your note package to the next page. You're going to see scientific drawings, and usually when we do a lab about microscopes, we teach you about scientific drawings as well, because it's not only about what you're going to be seeing through the microscope, it's going to be what you're communicating as well. So you should be able to look at what you see in the microscope and draw it on a sheet of paper. There are some rules for that, which I'm going to let you read and understand by yourself. Mainly what I want you to take away from it, it's not art class, right? So you're not looking for the prettiest picture. You want it to be clear. You want it to communicate. It has to be labeled really well so that you can communicate what you're seeing in the microscope to somebody that's just looking at your paper. So I'm going to let you do that by yourself, and that's pretty much the lesson. Bye.